Let's start with a few facts about developing brain. So the human brain has about 100 billion neurons. That's about more than 10 times the number of people living today on the entire Earth. So a large number, but they're not independent. They form circuits, networks, that are connected by 150 trillion synapses or connections. So it turns out the brain has all of the neurons, virtually all of the neurons that it will ever have by the end of the second trimester of pregnancy. And to get to that large number of neurons, at the peak production period, neurons are produced at 250,000 neurons per minute, or 15 million neurons per hour. The brain is about 25% of its adult size at birth, and then it goes to about 95% of its adult size by 6 years of age. So what happens over that postnatal period? Well, this is a picture of microscopic slides of the cerebral cortex, the cognitive processing part of the brain, at 1 month, 6 months, and 24 months, 2 years. Each of those little black dots is the cell body of a neuron. And you can see that something changes over time. The density decreases and there's more space between the black dots. And what neurons really look like are trees. They have what are called dendrites, and the dendrites increase in number and density in that early developmental period and form the connections that I was talking about. Now, this is such a complicated process that it's not surprising that every once in a while there's an error in the program of brain development that ultimately changes the structure or function of the brain and leads to altered behavior. And that's what we think are the causes for some forms of autism. Now, you can chart the course of brain development using techniques like magnetic resonance imaging. And you can see these pictures. That's what these pictures are, going from 1 week to 10 years as the brain gets larger and larger. But magnetic resonance imaging, which we do a lot of at the Mind Institute, my own research, is like looking at the Earth from 30,000 feet. You actually can see the big picture, but you can't actually see the cellular and molecular details of brain development. And so to answer why study the post-mortem human brain, there's a number of answers to that. Sorry. First and foremost, if you do histological... Is that something I'm doing? Oh, sorry. Do you want me to use these instead? Okay. Okay, now here we go. So the first answer is that if you do post-mortem studies, you can look at those neuronal networks that are responsible for function, for behavior, and see if there's any alterations in those. But as Matt State was just saying, another reason for doing post-mortem studies is that you can apply advanced genomics and proteomics techniques to look at the molecular biology of the autistic brain. Now, really, there are myriad mysteries about what's different about how the autistic brain develops that we don't at this point in time understand. That analysis of post-mortem brain material may give us some insights. So one example that I'm particularly interested in is these are 3 images of the brains of children at 3 years of age. The one on the left is a neurotypical child. The one in the middle is a child with autism. And the one on the right is another child with autism, but has a particular big brain form of autism that we see in 15% of boys with autism. Now, it turns out that this 15% of boys that have the big brain form of autism typically have the profound form of autism, where they have low language function, lower IQ, and a very poor prognosis developmentally. And even though we can chart with MRI that the brains are larger, we don't know at this point in time why the brains are larger. This is something that we hope we will be able to do by analyzing post-mortem tissue. Now, this isn't the case for all brain disorders. Over 100 years ago, Alois Alzheimer studied the brain of a woman who had a presenile dementia. She was a young woman who ended up with a dementia. And he found that in her brain there were these peculiar profiles. And subsequently, this has been studied for the last 100 years, and we now know that the plaques and tangles are the hallmark features that provide a diagnosis, a definitive diagnosis of Alzheimer's disease. And more importantly, by studying what constitutes these plaques and tangles, virtually all of the research that's going into treatments and preventative measures is based on our understanding of the post-mortem appearance and biochemistry of these plaques and tangles. So post-mortem research directly led to current treatment efforts. And this is a paper that just appeared at the end of last year. I think that this is actually sort of a remarkable testimony to the value of post-mortem research. What this paper found, in short, was that there are people who, when you analyze their brain, they actually have plaques and tangles, but they don't have dementia. And the question is, why don't they have dementia? Well, these scientists in these papers found that there were preventative mechanisms in the brains of these fortunate people that did not allow the plaques and tangles to degenerate the brain and lead to the dementia. And one would hope that as more research is done and we harness the information about what leads to these preventative measures, that we can use this in more and more people, not the lucky few that actually have the non-dementing form of Alzheimer's, but in everybody. And again, this is an example of where post-mortem brain research is leading directly to interventional strategies, or could. Now, the same is not true for autism. Autism has only been studied actually for a very few years. The first studies of post-mortem studies of autism were done by these two pioneers, Margaret Bauman and Thomas Kemper, and their first paper came out only in 1985, so relatively shortly ago. And what they found, they didn't see any hallmark features like plaques or tangles at this point in time. They found some changes in the brain, and some of these have been replicated, but more work needs to be done. So I wanted to give you just two quick examples of what has been found based on post-mortem research about autism. So I mentioned that we have 100 billion neurons, but they're not all alike. So there are some, like the green one in the middle, that are excitatory neurons. When they communicate to the next neuron, they actually activate it. And there's some, like the blue one in the lower left, that are inhibitory neurons. They actually damp down activation. And if you have an imbalance between the green ones and the blue ones, you actually have diseases like epilepsy, or you can have. There's a prevailing hypothesis in autism that a lot of the behavioral symptoms are actually due to an imbalance, again, between the excitatory neurons and the inhibitory neurons. Well, there's evidence for this. This was a study published by Verona Martinez-Cerdeño, who's at UC Davis. And basically, what she found was that one type, the little blue one, cell on the lower left, that are called chandelier cells because their axons look like chandeliers or provide something that looks like chandeliers, there's fewer of those in the autistic brain, suggesting that there's less inhibition, more excitation. This could lead to all kinds of behavioral manifestations like hypersensitivity to sound or to other sensory stimuli. In the realm of genetics, we know that if you have a mutation that affects the sperm or egg, it will affect all the cells in the body. That's called a germline mutation. But what is also known is that you can have mutations somewhere down the development of an organ. For example, in the brain, as those neurons are replicating rapidly, there could be an error in the DNA replication leading to a local mutation, and that's called a somatic mutation. Well, work that's coming out of Chris Walsh's lab at Children's Hospital in Boston has shown, based on post-mortem analysis, that individuals with autism have increased numbers of these somatic mutations in neural enhancer sequences. These are sequences that determine whether a gene is going to be active or not. Again, very early days, but this would suggest that a genetic mutation that you might see in the blood, or that you might not see in the blood, you would see in the brain if you analyzed it, and that mutation in the brain, a very focal mutation, may actually be causing some of the symptomatology of autism. Bottom line, we do not have enough post-mortem brain tissue to be able to carry out this state-of-the-art research. And for that reason, we approached the Simons Foundation in 2013 with the prospect of setting up a network of collection sites in the United States that would lobby the autism community to consider making post-mortem donations. I know this is not an easy topic to talk about, but the goal was to produce a critical mass of post-mortem donations that we could then distribute to autism researchers around the world who could do this state-of-the-art research. I won't go into details, but I would say that since 2014, when we were formally established, we have now collected 400 cases of both autistic individuals and matched non-autistic individuals so that we can do comparisons. And we've been distributing tissue quite literally all over the world, heavily within the United States, but also to Australia, to Spain, and to other countries. So I just wanted to, the last couple of slides is, how do you make a donation to Autism BrainNet? Well, Autism BrainNet has a staff that tries to make the process of doing brain donation as simple as possible. And we treat every family who wants to make a donation with compassion and with respect. So the first thing you do is we have an 877-333-0999 number. This is operating 24 hours a day, 7 days a week, 365 days a year. If a donation is imminent, we can call that number. And number two, our clinical staff will actually walk the family through making a brain donation. Number three, after the donation is made, and we have collection sites in California and in Texas and in Massachusetts. So we actually can have the donation go to the closest, we call them nodes, collection sites. We also have one in Montreal, Canada that's just getting underway. But then our clinicians will reach out to the family to get information about the individual, to confirm the diagnosis and get more information about what their characteristics were. We have resources for families. Making a brain donation is not like making an organ donation. If you have an indication on your license that you want to make a donation, that actually doesn't count for the brain. You have to let your intentions be known that you want to make a brain donation. So we have a brain donor card. All this can be found on our website. We have two of our outreach staff here, Cece and Lilium. Where are you? Let's see. Okay, yeah, here, right in front. Okay, if you would like more information, I'm almost done here. If you'd like more information, please contact Lilium and Cece. We have an aftercare program where we continue communicating with the families. We provide them information about how their donation has been used and the kind of research that's being done. All of these folks that you see here, we have on our website a Memories of Hope page, where many families, all of these are donors, families will provide testimonies about their family member. Some of them are difficult to read, but they all provide evidence that the families did this in an altruistic way. They thought that it was going to provide a path forward for more sophisticated research. Most of the families that we interact with say that in a trying time, making a brain donation actually gives them some comfort. And I wanted just to end with the words of one mom who made a donation, and you can listen to the reason that she had for making this donation.
Declan was my 6-year-old son. He passed away due to a seizure in his sleep. He loved music. That was one of his big passions, was anything music. He loved playing his keyboard. He loved playing the guitar. And then he also loved books. He was the light of our life, and he was so kind and precious and sweet and just a ball of sunshine. There have been studies that have shown that kids with his diagnosis have passed away in their sleep. It's called SUDIP. And that's unfortunately what happened with Declan. So I decided to donate to Autism BrainNet because my husband and I wanted to help others that were in the situation that we were in as a family. We were hoping with his tissue donation that this would help research for autism and help others discover ways to help individuals with autism, maybe different therapies or maybe different, medical advances, anything that could help and honor Declan at the same time.
Can you please join me in thanking all of the hundreds of families that have made donations to Autism BrainNet?